Vale, ya está conectando. Perfecto. So, welcome everybody. I'm gonna silence. Yeah. Welcome everybody to the Bionanosurf Scientific Seminars. This is going to be our, not first, but yeah, this is the first transmission in YouTube of our scientific seminars. We have today here with us Professor César de la Fuente from University of Pennsylvania, who's going to talk about the antibiotic discovery by means of computers. And Scott will present the conference today.
Okay. Hey, thanks very much for joining us, César. It's a real pleasure to have you. So Professor César de la Fuente, he is, since last year, the Presidential Assistant Professor at the University of Pennsylvania in Philadelphia. And as Elena said, he leads the machine biology group, who work to develop new computer-made tools and medicines to replenish current antibiotic arsenal, and also develop new low-cost diagnostic technologies for infectious diseases. César is an NIH, National Institute of Health, MIRA investigator and a BBRF Young investigator. And also he has an impressive list as well as one of the world's top innovators for digitizing evolution to make better antibiotics. That was recognized by the MIT Technology Review last year. And also, especially in the last year or so, he's been the inaugural recipient of the Langer Prize in 2019, ACS Kavli Emerging Leader in Chemistry this year, 2020, the Nemirovsky Prize in 2020. And also he received the ACS Infectious Diseases Young Investigator Award in 2020. In addition, he was named a Boston Latino 30 under 30, a 2018 Wonderkind by Stat News, top 10 under 40 in 2019 by Jen, among many other prizes. And his research results have yielded more than 75 peer-reviewed publications and multiple patents. So yeah, César, it's a real pleasure to have you here giving the talk today. So thanks very much for accepting.
Yeah, thank you. Thank you, Scott, for the kind introduction. And thank you, Elena, Scott, and Jesus and company for coordinating this webinar and for the invitation. So today I thought I would tell you a little bit about what we've been doing in the lab in trying to develop novel classes of antibiotics using computational tools. The motivation behind all these efforts is that really we're running out of antibiotics, and antibiotics are the foundation of modern medicine. They protect us from infections stemming from injuries and childhood maladies, and they make all surgeries, transplants, and childbirth and chemotherapy that we use in the hospitals every day possible. And antibiotic-resistant infections are predicted to become the leading cause of death in our society. They are estimated to kill 10 million people annually by the year 2050, surpassing even cancer as a cause of death in our society, as we can see here, costing the global economy $100 trillion and corresponding to the staggering number of one death every 3 seconds. So that's where we're heading. And so that's why we need to come up with novel antibiotics. And really the natural world, right, has been a great innovator in terms of its ability to supply us with novel antibiotics, but it's really running out of inspiration. In the last decades in the last several decades, we have been unable to discover any truly novel antibiotics in the natural world. And so my lab believes that the next generation of these drugs is going to come most likely not from nature, but from machines. And so what we're trying to do is we're trying to encode all these chemistries that exist in the natural world into ones and zeros, so then computers can take care of the discovery process. And so the next question is which subset of molecules should we focus on to encode onto computers to carry out this innovation. And really, if you look through the tree of life at all the different potential molecules that have intrinsic antimicrobial properties, there is a subset of them that have evolved to have these antimicrobial activities. And they belong to this family of small proteins called host defense peptides, or HDPs for short, which are very small, typically between 10 and 50 amino acids in length. They're genetically encodable because they're peptides, small proteins. They're chemically synthesizable, and they therefore represent excellent scaffolds. And again, like I mentioned, their production throughout nature is widely conserved. So bacteria produce these molecules, and we produce these molecules as part of our immune system. So the next question is, how do we engineer these HTPs? And so in order to do that, we follow very basic principles from protein engineering that tell us that if we control the sequence of amino acids, which are essentially the building blocks that compose these peptides or proteins, we'll be able to control their biological function. And so I liken this to really playing Lego in the laboratory. That's what we do every day. We try to arrange different amino acids in certain positions to reprogram the original molecule so we can have new synthetic versions that are more effective. And more specifically, we use a convergence of principles that we call machine biology from different disciplines. So including computer science, synthetic biology, infectious diseases, chemistry. We bring them all together to try to tackle this big problem of creating novel antibiotics. And this is the general framework that we use in the lab to truly understand these molecules. We first want to understand from first principles the underlying chemistry and physics of these molecules. Next, we're in a position to control their structure function, both chemically and biologically, as I'll explain throughout the talk, in the laboratory. And finally, once we've achieved a good level of control in the lab of these molecules, we can encode them onto computers so they can take care of the innovation and discovery process, ideally without much human intervention. And I think the first two steps of understanding and controlling are key, because otherwise it would be impossible to teach a computer something that we humans don't fully comprehend. And so I'll talk about each of these vignettes today during this talk. And the first one is to try to understand from first principles these molecules. And I'll tell you a couple of stories around this. So the first one is the following. So we took this peptide molecule derived from the venom of this wasp. And this molecule has a very interesting history because when it was first isolated years ago, it derived into a lot of excitement because people thought they could develop it into a new antibiotic because it was a good molecule at killing bacteria. But the excitement really dropped when scientists figured out that it was also a very good molecule at killing human cells. And at the time, there were no good tools to try to reprogram or reverse that activity, that deleterious activity. So as bioengineers in the lab, we took this molecule and we challenged ourselves and asked the question, can we reprogram this highly toxic molecule that is a promising new drug, but is too toxic? Can we reprogram it into something that is no longer toxic, but preserves or even enhances those intrinsic antimicrobial properties? So again, just long story short, we played Lego with the molecules. We tried a number of things, including high density mutation screen of the wasp venom molecule, with the idea of filtering out those amino acids in specific positions that were associated with toxicity and also antimicrobial activity. So we're trying to tune those two biological functions. And things that we looked at, just in summary, we looked at physicochemical descriptors, like I mentioned, the underlying physics and the underlying chemistries. We looked at things like secondary structure, hydrophobicity, net charge, etc. And then we created mutants in the lab, and we tested them against clinically relevant bacterial pathogens that we can see here. And basically, we got a lot of different color schemes here, which represent essentially different abilities to kill the bacteria. So some of the mutations enhanced the ability to kill bacteria, and some of them decreased or depleted that ability. And so that allowed us to really learn a lot about these molecules. And so things that we learned, for example, was, so this is the wasp venom molecule. This is how you typically draw it on a piece of paper. So we can see that it has these two distinct planes, this one here, and then this one here. So things that we learned, for example, from this subset of experiments is that if you mutated these amino acids here indicated by these blue arrows, that led to a loss of function. And conversely, if you mutated these amino acids here indicated by these red arrows, that led to enhanced biological function. So we learned all of this, and we were able to even correlate those changes that we made to the specific physicochemical parameters, such as alpha helical propensity, hydrophobicity, etc. And so once we know what to change in order to affect biological function, we can easily tune those molecules in the laboratory to have optimized activity. And so that's it leads us to the second step, right? To be able to control in a predictable manner these molecules in the laboratory. And really, the concept here, like I mentioned, brings us back to Feynman's words of what I cannot create, I do not understand. And this is really aligns very well with this case, where if we cannot really create these molecules in the lab, we don't fully understand them from basic principles. And so that's what we did. We took all those things that we learned, all those rules that we learned in the previous experiments, and we were able to optimize the biological function of these molecules, of these venoms, to create a highly active antibiotics. And in the process of doing so, we were able to isolate also non-toxic variants, which was very exciting. So we were able to reverse the deleterious toxicity of the venom. And then we, in my lab, we're very interested in always trying all the molecules that we create in preclinical models, mouse models, because we're very much interested in translating everything we create into potentially, hopefully, the clinic. That's the overarching goal. And so we developed this mouse infection model, and we tested the molecules derived from the wasp venom to see if they could resolve infections in this context. So here we have the initial set of data where, again, the red peptides, which are the mutants that led to increased activity, we can see that they resolve the infections better than the blue peptides that are the ones that lost function. And in black here, we have the untreated control group of mice as a reference. And these are very low doses of each of those peptides. But what we did then is that we took the best one, we increased its dose to 64 micromolar. And what we observed here was quite remarkable. So after 4 days, it was capable of sterilizing completely the infection, which was very exciting because that's not typically something easy to achieve in the context of this mouse model, and at least in our previous work with this particular mouse model for years. So I'll tell you now about another project similar, where we started with a venom again from wasp in this case, but we started with a computational from a computational perspective. So we ran a large computational screen to try to find motifs in existing molecules that associated with high antimicrobial and immunomodulatory activities. So we found this motif composed of these 5 amino acids that was highly conserved. And then what we did is we took this peptide from another venom, and what we did is we engineered in the motif into the original sequence of the venom, creating the synthetic molecule. And just briefly, that change allowed us to incorporate these two activities, antimicrobial and immunomodulatory activity, into that molecule. So generating a molecule with dual activity that was capable of boosting, so it was capable of resolving infections both by directly targeting the microbe and eradicating the microbe, and indirectly resolving the infection by boosting the immune response. And in addition to boosting the immune response, it was also capable of controlling inflammation And this is important because whenever you have an infection, you obviously want some level of inflammation because it allows you to clear the infection. But oftentimes, that inflammatory response can become exacerbated, which can lead to sepsis. And sepsis can literally kill you. It can be extremely lethal very quickly. So what we're able to show here is that this molecule or subset of molecules that we generated was capable of, like I'm showing here, resolving infections in mice against both laboratory strains and clinically drug-resistant strains through its dual activity of directly targeting the microbe and boosting the immune response while dampening or balancing the inflammatory pathway. But again, just to finish this part, I've shown you how we can use mostly chemical methods to essentially understand and also control these molecules in the laboratory. And as an example, I've given you two subset of projects where we've done this to repurpose venoms into novel drugs. But we can also control these molecules biologically, not only chemically, because as I mentioned, they're genetically encodable. So we can essentially encode them in genomes. So then we can utilize living organisms as factories to produce these molecules. And you may want to do this for a number of reasons. So one of them, to lower the cost of manufacturing of these molecules. You can also encode them in plasmids to create high order libraries to create a lot of these different variants. And another example would be to come up with living medicines. And the idea here is that you can take bacterium that grows in your yogurt, you can engineer it to produce some of these molecules. And then the idea is that you would take the yogurt in the morning with the engineered microbe, and it would go through your body, through your stomach. It will reach your gut, it would colonize your gut, and then it would secrete these molecules in real time so you can have a therapeutic output. And these are all things that we're exploring in the laboratory. But for the purpose of this talk, I'll talk about the manufacturing approach. So in this case, we use yeast as a producer as a sort of like a factory of these molecules. And this is the vector architecture that we use. And this is the overall sort of scheme. We use this carrier protein called human serum albumin, or HSA, to provide or confer stability to the complex. And then here in blue, I'm showing the peptide sequence. And again, this is just amino acid code. You can incorporate any code that you want, any peptide sequence that you want. And then we engineered in here a cleavage site that allowed us to cleave off the peptide once we've produced sufficient quantities of it. So this is the general engineering strategy. You have the PK genome. So PK is a type of yeast. And within the genome, you incorporate a recombination site, which is this red triangle. Then separately, you have this plasmid, which is a circular piece of DNA that also has a recombination site in blue. And then it has a promoter that essentially promotes the expression of whatever it's downstream of it. And downstream of it, we have a peptide of choice. So what happens when you add to the reaction, so basically you have this in a test tube and you add to the reaction an enzyme called recombinase, which is this little cloud here. What it does is it allows recombination of these two sites, therefore allowing integration of the plasmid into the genome. So then essentially you get a very robust production of the peptide from the genome of the yeast. And then this is just the general workflow, how we did it, and the experimental evidence that by the end of the experiment, you have your peptide separated from the fusion protein. And in each case, of course, we make sure to validate the antimicrobial properties of all the peptides that we generated in this fashion. Of course, the next question is, can we scale this up? Can we really decrease manufacturing costs of these molecules? And so what we did is we scaled it up in a bioreactor, the system. And the cool thing is that the promoter that I mentioned as part of the architecture, the vector, that promotes the expression of the peptide can be easily induced by adding methanol into the medium. And so if we have the bioreactor here, we have the yeast growing. If we add methanol, we see an increase in the production of the protein. And again, this is the same, essentially, upon addition of methanol over time, you see increased production of your protein and peptide of choice. And so how does this relate in terms of cost? And so we ran a number of preliminary calculations for this, and essentially using traditional methods for synthesizing these molecules, which is essentially a solid phase chemical synthesis. To generate 1 milligram of peptide, you need between $100 and $1,000. Using this yeast system, you can come up with the same amount, 1 milligram, for less than $1. And I have to say, with one caveat here, this is not considering purification costs, which would add additional costs to that. But nevertheless, a huge decrease in the cost of production of this molecule. So we're excited about this possibility and looking to scale this up even further. So I told you how we can, we can understand these molecules now in the lab quite well. We can control them both chemically, and now I've shown you just in the last couple of slides how we can do this biologically as well. So we're really in a position now to encode all this information, all these rules that we've learned onto computers. We're in a good position to do so. And so that's what I'm going to tell you about next. Proteins and peptides are, as you know, composed of 20 amino acids. Well, those are only the naturally occurring ones. We can also incorporate non-canonical ones in the lab. But this confers them with a huge chemical space. And one of the things computationally that we can do to be able to discover novel antibiotics is to explore this chemical space. And so just to relate to you how vast it is, I'm giving here some sequence space comparisons of concepts that you may be more familiar with. For example, the number of people on Earth is 10 to the 10. If you keep going higher and higher in terms of orders of magnitude, the number of stars in the universe is 10 to the 31. Well, if you consider a very small peptide, very small protein, composed of only 25 amino acids, again, this is much smaller than any protein that we would be creating in our bodies, which are composed of hundreds of these amino acids. That gives us a sequence space that is superior to the number of stars in the universe. So clearly, we need computers to be able to exploit or explore these amazing subset of possibilities. And in addition, to make things more complicated and interesting, biological evolution through billions of years has only sampled a tiny fraction of the entire sequence space of possibilities of all potential molecules, indicated here by all this white space. So what we wanted to do here, we wanted to expand the boundaries of sequence space to be able to use computers to reach all this white space here, to be able to find new molecules with biological functions that are different from what exists currently in nature. And so clearly we need computers. And then the next question is, how can we teach computers to innovate these molecules? And we ended up reaching the conclusion that the best way to do so was to use the same principles that biology has used for billions of years to evolve and innovate. And that is, of course, utilizing Darwin's algorithm of evolution. So we essentially decided to train machines to execute this algorithm to be able to innovate in the antibiotic space. So this is how we did it. We took an initial population of the HDPs that I mentioned from the natural world, and then we trained the computer to perform this mutation selection recombination, which is essentially a theory of evolution in n-fold iterations in a feedback loop to be able to generate diversity. And here we can see one of the simulations. So over different iterations, we can see how the computer evolves the molecule towards optimal fitness values that correlate with predicted antibiotic activity. In the process of evolving the molecule to optimize it, the computer is also capable of exploring previously unexplored regions of sequence space, so the white space that I mentioned, to yield molecules that are different from what we typically see in the natural world, which is also incredibly interesting. So here I'm showing computational simulations of some of the best molecules created by the computer. At this point, we reach a roadblock that is common to really all innovations involving computers. And so all molecules created by the machine represent just a prediction, like I mentioned. In other words, the machine thinks that the molecules it has created will be good antibiotics, but we still need to validate this in the lab experimentally. So that's what we did. We synthesized some of these molecules, and we tested them in the lab against clinically irrelevant bacteria. And this is what we found. So we found a lead molecule that needed a very low dose to kill bacteria. And particularly if we compared it to the templates that the computer started simulation with, which were essentially not very good antimicrobials. And we did this on purpose to show us a proof of principle that we could train a machine to convert a non-antimicrobial molecule into an active one. So to sort of reverse the activity. And then this is the three-dimensional structure of the lead molecule found by the computer. We can see that it forms this beautiful alpha helix. And this was elucidated by NMR. And then this is the same structure in three-dimensional space. Now, again, like I mentioned, we're always interested in translating everything we do. And so we asked the question of whether this machine-made molecule could resolve infections in a mouse model. And here are the results. So basically, the machine-made antibiotic resolved infections better than the templates that the computer started the evolutionary process algorithm. And then here we have the treated control group of mice as a reference. So you can see here the number of bacterial cells or colony-forming units per milliliter that we have in the treated control group of mice. And so we're very excited, obviously, about this. This is really the first demonstration that we can train machines to create antibiotics that are not only useful in vitro in the lab, but also useful in a preclinically relevant mouse model. And in addition to using computational tools, excuse me, to design novel antibiotics, like I've shown here, we're also interested in using computers to discover novel antibiotics in perhaps places where people have not traditionally looked before. And the truth is that if you look at biology and biological code everywhere, genomes, proteomes, there's a lot of previously unrecognized information that has really not been explored very well. And so the idea here is how can we decrypt a lot of this information to be able to unveil this treasure trove of novel potential interesting molecules and potential medicines. And because of all the previous work that we've done over the years and some of the work that I've presented today, we knew which molecules to look for, so we could now mine biology searching for such molecules. So to address this problem, we took inspiration from image and speech recognition algorithms, but instead of recognizing facial expressions or sounds, we wanted to recognize molecular patterns encoding antibiotics. The concept is really very similar to having let's say you have a large Word document on your computer and you're trying to find the word hello within the document. So you know what the word hello is, you know the sequence of letters next to each other. And so it's very easy to find it. So this is kind of the same concept. We knew what to look for, we knew how to find it, and now the idea was just to mine biological systems through this search engine to really find novel antibiotics. So this is essentially how the algorithms work. If we have an entire molecule, in this case a protein in 3 dimensions, we can also display it in 2 dimensions. And what the algorithm does is it runs through the code of amino acids and it finds regions within the code that have predicted antibiotic activity, and those are given by these different color schemes here. This is all directed by an antimicrobial score, or AS. And of course, again, here, because we're using computers, the information obtained is just a prediction, but it's a very powerful one because it allows us to identify fragments within the entire molecule, here shown in yellow, that we can then extract from the molecule and we can play around with in the lab to develop potential new drugs. So using these algorithms, we have now mined the entire human body and have found tens of thousands of previously undescribed tiny molecular fragments hidden within the realm of our bodies that we refer to as encrypted antibiotics. And we've now synthesized a lot of these in the lab, and we've confirmed their antibiotic properties and even shown their anti-infective efficacy in mouse models. What we find fascinating about these findings is that they serve to connect the innate immune system to other systems throughout the body, including the nervous system, gastrointestinal system, cardiovascular system, because we found these encrypted molecules all throughout the body, not just in the arm of host immunity. And so we're very excited about these findings. So some of the things that we're able to show with these encrypted antibiotics is that if you take several of them derived from the same biogeographical area throughout the body, they're able to potentiate each other's antimicrobial activity. In other words, through synergistic interactions, they're able to really enhance each other to kill pathogenic microbes to even nanomolar range, which is within the range of concentration at which these molecules are physiologically produced. And another thing that we found quite striking is that the bacteria, such as Acinetobacter baumannii, which is a clinically relevant bug, they don't really develop resistance to these molecules. And here as a control, I'm showing a polymyxin B, which is a molecule used in the clinic, against which bacteria do develop resistance substantially after 25 days or so. So this is also quite interesting. Here I'm showing just 4 examples of 4 encrypted antibiotics that we found, and we can see that they're produced at different levels throughout the body in different organs. So we can see, high levels, etc. So this is just to give you a sense of the fact that these molecules are really produced physiologically. And then, of course, we tested the anti-infective activity in a mouse model. And we were able to show that, not for all the tens of thousands that we found, but for some of them, a subset of them, we're able to show anti-infective activity in monotherapy and also in combination therapy, again, through those synergistic interactions, which was very interesting, against clinically relevant microbes. So another subset of molecules that we found in the human body are produced in the human stomach. Essentially, we're able to find that these encrypted molecules were able to kill foodborne pathogens such as salmonella, which of course infects the stomach, both at a neutral pH and low pH levels that are relevant to the human stomach microenvironment. And we're able to show that also the anti-infective properties of the encrypted molecules compared to the treated control group of mice. So this is really a project that we're we have a lot of work to do. We have tens of thousands of molecules to explore, and we're doing that actively, so stay tuned. In addition, these algorithms can also be used to explore genomes of other organisms. They're not limited to the human body, of course. It can be extrapolated to other systems. And now we've been doing for some time with collaborators a side project where we're trying to look at, explore extinct organisms as a source of an previously untapped source of novel antibiotics. We've been doing some work with the woolly mammoth, which has a partial genome that has been elucidated, and we've been able to find interesting molecules encoded there that we're trying to explore in the lab. We can also use these algorithms to essentially analyze our own data sets that we create in the lab. So here in this case, we ran a large peptide screen against bacteria, by the end of which we had the amino acid sequences of each peptide and their corresponding antimicrobial scores. So then what we can do is we can run the algorithm, and the algorithm identifies patterns or regions that are associated with antimicrobial activity, similar to what I've shown before. And then we can use all that information to design a second-generation library that we can synthesize in the lab, and we can test against bacteria. And here, basically, what I'm showing is like the different hits that we obtained. And in black, we can see the molecules that we created that led to 100% killing of those particular microbes. And once again, we're able to correlate the in vitro data with the in vivo data. And so the best molecules here resolve the infection better than some of the molecule that was not so good. And here we have a treated control group of mice as a reference. So just to wrap up, I've told you how we could use computers to both design and discover novel antibiotics. And we're currently also interested in developing machines that autonomously create these drugs in real time. So the idea here is that you have your computer to both design and discover new drugs. Once it's created those molecules, it can transmit that information through Bluetooth to a synthesizer that will make the molecules the machine tells it to make. And then we can streamline that with a robot that will screen the molecules for antibiotic properties. Now, finally, that information can then be transmitted through a loop or loop back to the computer to have a self-learning autonomous system for drug discovery. And that's really kind of the dream that we have in the lab. And with that, you know, I'd like to thank the lab. It's really a privilege to work alongside them every single day. I'd like to thank our funders for allowing us to do the work we do. And here is the link to our website, my email address in case you want to reach out to follow up or collaborate, or if you have any questions or anything. And here's our Twitter handle in case you're in social media. And with that, that's all I have for today. So thank you for your attention. I'll be happy to take any questions you may have.
Thank you, Cesar. It has been really interesting to see you. So we will start with the questions now. So if anyone has questions, you know, I will. Okay, Hector.
Hi, Cesar. Thanks for the talk. It was really impressive. I have a couple of curiosities regarding the algorithm that you mentioned. You use the Darwinian evolution algorithm, but it is possible to use other algorithms like randomly explore the peptide sequence to search in order to search new sequences.
Yeah, sure, absolutely. Yeah, I mean, you can use other types of algorithms. You can use different classifiers to be able to classify antimicrobial activity and relating to structure or primary sequence. In this case, we selected this one because we thought it was a great way to be able to evolve the molecules, which we thought was very interesting. And the way we did that was using that fitness function that I showed, which essentially selected for minimal structures that interacted with bacterial membranes. But we're now thinking about incorporating other descriptors into the fitness function, including things that select against potential cytotoxic sequences, etc. So it's sort of plug and play, and it allows us to really look or evolve molecules that we really want. But absolutely, you can also use other algorithms. We're now using machine learning to try to achieve some of these same goals.
And another question is that usually antimicrobial peptides are designed to be a balance between positive charge and hydrophobic residues.
Yeah.
But it is possible to search for anionic antimicrobial peptides that I think is an unexplored chemical space.
Yeah, that's a very good point. Absolutely. I mean, in the particular computational work that we've done, we focus on prototypical sequences. So, like you mentioned, you know, with a certain ratio of hydrophobic to cationic composition. But I don't see why we would be limited to that. That's only the work we've done so far. But I completely agree that searching for anionic sequences would open up the sequence space in some way.
Thank you very much. Thank you for the presentation.
Thank you.
Okay, so we have Gabi as well. I will activate your sound.
Hi, Can you hear me well? Because my mic is kind of moving around. Okay. Thanks a lot for the presentation. I really enjoyed it. I was having a conversation with a friend of mine who is working with machine learning, and he was telling me that all this machine learning actually needs to be mostly outsourced in terms of computational power, because the private industry who actually has all this computational power to be able to manage all these very heavy algorithms. Do you have any idea how can what is the most interesting part that would attract this private industry into reaching a final product, or something that could be actually applied? Because I guess that's what they are mostly interested in, right?
Yeah, no, for sure. I mean, our main goal is to actually create, like I mentioned, algorithms that autonomously create new drugs that can be incorporated into the clinic, into phase 1 clinical trials. So that's really the goal. We have a grant that we were just awarded a couple months ago, and the main goal is that to basically almost without human intervention. I mean, at this point, it's impossible to not have obviously human intervention, but minimizing it to the maximum to be able to design computationally molecules that represent drug-like compounds that you can use directly. I mean, I would imagine private companies, startups would be interested in such a technology. But in my lab, particularly in terms of computational power, we have a number of workstation and nodes, and we also use supercomputer time in the US to allow us to do
that. Okay, thank you.
Thank you. Okay.
So next one is going to be Scott asking. So whenever you want, Scott.
Hi, Cesar. That was great. That was really interesting. Thanks a lot. I've got a few questions. Just the first one. You say you're using all these algorithms for computer guided antibiotic discovery and stuff, but then in the end, I assume, maybe I'm wrong, that you have therefore then lots of compounds that you're screening in the lab, doing inhibition assays in the laminar flow hoods. So is this your current limitation for really screening these molecules that are the most active?
That's a good point, Scott. Yeah, we're still limited by the need to validate everything experimentally. So that is kind of a bottleneck in terms of time and cost and labor. At this point, we are, but we're hoping that we build sufficiently large data sets in the future where this can be more automated. So we can have more predictability in terms of being able to anticipate cytotoxicity issues, solubility issues, antimicrobial activity issues, etc. So that's really like more down the line. But at this point, we are limited by we validate everything experimentally, including toxicity, hemolysis, like I mentioned, everything else.
But it really is like quite conceivable, isn't it, to have like a fully automated lab screening these compounds? Is that right? Yeah.
Conceptually, it definitely is. I mean, it's just a matter of building up data sets. Now we're using machine learning algorithms to be able to allow us to learn from those data sets, and we're creating everything in the lab from scratch. So under standardized conditions to be able to have, relatively homogeneous data sets that we can rely on. And so that's what we're working very hard on. But that's all experimental work that we then feed into the computational side of the lab.
That's great, thanks. See, can I ask another quick question as well in relation to your first part, your chemical approaches? Maybe you said it and I just missed it right at the beginning, because there was a lot of information in your talk. But where do you get your wasp venom molecule, or your was it like a frog venom molecule? Is that something that you just purchase?
Yeah, so some of we synthesize them in the lab, but some of them were previously described in the literature. So we didn't necessarily discover those as new sequences. The ones that we did discover as new sequences were all the ones that are from the human proteome. Those are all new.
Okay.
But the wasp venoms were previously described in the literature, essentially, and we then we then synthesize them chemically in the lab, and we start playing around with them.
That's cool. Is that Marcelo, who's is one of the guys that's synthesizing these? Yeah. Yeah,
Marcelo is the one leading leading a lot of things, including the chemical side of things of the lab. Yeah.
Okay, that's cool.
Okay.
I see that there's lots of other people raising your hand, so I'll speak to you again later. Thanks a lot.
Thanks, Scott.
Perfect. So now, Susa. Okay.
Thank you. Thank you for this very interesting presentation. I have a question about the in vivo stability of the peptides. That is generally an issue with the peptides using for treatments. And do you have any process to incorporate this issue in the design of the peptides, or for example with not natural amino acids or D amino acids?
Yeah, Susan, that's a great question. So for the computational work so far, we've been limited to linear sequences. So as you mentioned, some of them, a lot of them might be susceptible to proteolytic degradation and things like that. Surprisingly, the encrypted peptides are not that susceptible to degradation, but some of the other ones are. And so what we've been doing so far is down the line, what we do is we tweak the molecules chemically. So one of the papers that we're about to submit now, what we did is we took one of these naturally occurring peptides that is essentially a linear sequence, and what we did is we created a retroinversa version with the enantiomeric amino acids, and so that obviously increases stability against proteases, etc., and allows it to resolve the infections much better. But with some of them, we're not really having a lot of issues. But in the ones that we do, we just tweak the chemistry, like you mentioned, either by cyclizing the molecule, constraining it, or creating disulfide bonds, or incorporating non-canonical residues. But a lot of the mouse models that I've shown are topical, so skin infections. So in those ones, we don't typically see we don't have any major issues with stability. However, when you go to systemic infections like the venom derived antibiotics that I showed in the sepsis infection model, in that case, you need to obviously worry a little bit more about that. But yeah, great question that we're thinking about actively, and we're starting to think about how we can computationally design antibiotic peptides that are not only linear. So we're starting to think about that.
Thank you.
Thank you.
Okay, so next one is Raluca to ask questions.
Hello, can you hear me?
Yeah, hi Raluca.
Hi, Cesar, thank you very much. It was really amazing. So I have a couple of questions. First one is a bit in line with Scott's comment about the current limitations that you have right now, that you have to validate everything in the lab and so on. So my question was if you try to use a kind of quality by design approach for the theoretical part. So like having a pilot set of data and taking into account some of these properties that your peptides should have, like biological activity, stability, whatever.
Yeah, I mean, we've done it in terms of antimicrobial activity only so far. So the fitness function, for example, in the genetic algorithm that we used that drove the evolutionary process, it essentially selected for sequences of amino acids that had a certain hydrophobic moment and alpha helical propensity, which essentially translates into positive amino acid, cationic amino acids and hydrophobic residues with some sort of motif. So just looking, essentially focusing on interactions with the bacterial membrane, because that essentially the positive charge allows the the peptide to interact electrostatically with the negatively charged membrane of the microbe, and then the hydrophobic domain allows it to translocate into the membrane. But we're now looking into incorporating other things like cytotoxicity, hemolytic potential, and things like that. So counter-select for toxic molecules as well.
Okay, and my second question was if you try to look for other types of antimicrobial or antibiotic structures such as glycopeptides or beta-lactams or...
Yeah, no, that's a good question. We haven't done that yet, but we are getting very interested in trying to find more cyclic, more molecules that would be more readily accessible to using humans. So cyclic structures, constrained structures, etc. We haven't really fully finished any of those projects yet, but certainly of interest.
Thank you. Yeah, it was very nice, and especially this part with the encrypted active molecules was really interesting. Thank you.
I'm very excited about that project and the follow-up projects coming from that. Thank you.
Thank you.
Okay, so I think those are all the questions for now. And yeah, we don't have any from the moment in YouTube. So I would like to join as well to thank you for your great talk. And it was really nice to have you in the seminars today.
Yeah, thank you so much for having me. And yeah, look forward to perhaps visiting you guys at some point or giving another talk at some point.
Yeah, we'll see how the year goes and the next year as well.
You're welcome to come to visit us whenever you want. And
we're all looking forward to the next year, right?
okay.
Yeah, it was on the cards for you to be over visiting this year. Let's see if next year we get the opportunity to invite you over properly as soon as the borders open up.
Yeah, I know. We'd love to. We'd love to go with you guys for a dinner and have some nice food for sure.
Thank you, Cesar, for your seminar.
Okay, guys. Take care.
Thank you.
Thank you, Cesar.
Cheerio.